Hello there, Year 10. Welcome to your next lesson to do with plant and animal adaptations. This is actually a field investigation. You will either be able to do it in your garden, in a park, providing you follow social distancing, or you can do it on paper if you are not able to or allowed to go out. You have got a calculator on this front sheet to show you that you will need one for this lesson, and there is a symbol whenever you'll need it during this lesson as well. So a number of slides have got this same calculator slide on. You'll do now on the front slide here shows a graph. If you have a look carefully at the axes, you will see that the y-axis, the one that goes up and down, has got temperature in degrees Celsius on it. So at the bottom it's low, on the top it's high, maybe 0, may not be, and on the x-axis going across we have got time in minutes. Now that means that this graph gives us two bits of information, temperature and time, and as something gets hotter over time, changes are occurring. There's points A, B, C, D, E and F. This relates to something that's being heated and changing, and I want you to work out what the labels are to go at A, B, C, D, E and F. Remember that you can pause the video here while you do it. Right, here are your answers. You should hopefully have got most of these. Obviously we're going to start with a solid, then we'll have a liquid phase, then we'll have a gas. Where these change are the plateaus, the straight bits. The temperature doesn't go up while a change of state is happening. This is a heating curve. We looked at these earlier this year. So A is a solid and it's being heated. At point B it starts to melt and we get a mixture of solid and liquid particles, but at this point the temperature does not change. At point C it is completely liquid, so if we were talking about the chemical H2O, A would be ice. This would be a mixture of ice and water. Here we would have all water. We'd be heating the water up as the water reaches the point at which some molecules are going to start to change from a liquid to a gas. That is point D, where we change from liquid to a gas. This is called boiling, and at F all the molecules are now gases. Molecules, still the same molecules, but they're in three different states: solid, liquid and gas. These are the changes. The changes only need to have boiling and melting. They do not need to have condensing on there or freezing, because we can see from the axis that it shows us that temperature is going up. Okay, so here are our objectives for today. We're going to understand why sampling is used. You need to be able to explain how to use a quadrat. This does come up in exams a lot. You need to get it right, you need to be able to explain it, and that is why we're going to practice it. You're going to have some practice calculating means and areas. Again, they come up a lot in tests, and you're going to be evaluating the accuracy of your results. There are two key words you need to know. You need to know abundance, which is how many of a certain organism are found in a certain area. So it's not just how many, it's how many in a certain size of space. And distribution is how they are spread around the area. Think about throwing sweets to your brother or sister. If you're accurate, there might be abundance. They might have a lot of sweets at their feet. If you're less accurate, they may well be spread out over the living room, and then you'd get a distribution of them around different parts. Now, here we are, picture of the school field, and why do scientists sample? Now, if I asked you to count all the plants or all the animals that are in this field, it would be an awfully long job, and it'd be very easy to make a mistake, and no one's going to have to want to go back to the beginning and start over. It's far easier to make a mistake with animals as well, because they move about. If you ever remember those big black crows on the school field, they're hopping about constantly. Trying to estimate how many would be really tricky, unless you took a sample at a certain time, perhaps by taking a photograph, and then you counted them. Plants are easier because they don't move about, but they are more common, therefore the numbers are bigger, so it's easier to make a mistake with the actual counting. When you take a sample, you either count a small area, what's in a small area, or you count what's involved in a short time span, and you use that information to estimate a total. An estimate is not a guess. A guess is just a random number. An estimate is based on evidence and some good mathematical workings. Now, there are different types of samples. If you want to catch and sample an animal, it's harder because they move. So one example where you can do this is something called a pitfall trap. So in here we've got a flower pot, one of those little plastic ones. The reason we've chosen a plastic one is because the sides are shiny and slippery, and so if a bug that's walking along the ground here, you've got to cover over it to keep rain out and to stop birds and things coming along and eating whatever you catch. Put it propped up round the corner, bug comes along, falls in, because it's a shiny plastic container, bug can't get out. And that will stay there until you come along, take off the lid and count what's in there. There's small holes at the bottom to allow drainage, so that if it does rain they don't drown the insects. That would not be very ethical. Don't want a big hole though, because you don't actually want them getting their way out of it. So this would work for small land living animals such as ants or woodlice, using a very small trap. Larger pitfall traps can be used for larger wild animals, but you would need to be much more careful doing that. This is something you could make at home, but do get permission from the adults who are in charge of the garden, and make sure you position it somewhere where people aren't going to disturb it or aren't going to fall into it. Another way to sample is by observing, that is watching. For example, you could count the number of birds visiting a bird table in a given time. And the RSPB, the Royal Society for Prevention of Cruelty to Birds, coordinate a national survey every year where people across the country on a certain day at a certain time count how many of certain birds that they see. The possible problems with a bird table sample is that only some birds will be attracted to the food you put out. Different birds eat different foods, and of course if you have a particularly large bird, it may not fit on the bird table at all. When we come to plants, it's easy because they don't move, so one way you can check is by having something like a transect. That's a line, and you take samples along it like this, conveniently numbered from 1 to 10. And often these are used if you've got a change from the beginning to the end. You might be going from a shady area next to a house out into the middle of the field to see the effect that brighter sunlight has on what's growing there. You could be looking at a beach, then this transect would be on a slope, and you could be looking at number 1 could be well above the sea level, and number 10 just goes below water when the tide comes in. There are all sorts of ways you can use these. But what we're going to use are Quadrat. You've seen these at school. At school we have big frames that are split into smaller frames. You're not going to be able to make those at home, but we have got a cunning plan, do not worry, all will be revealed later on. So a Quadrat is a square frame that is used to sample a small part of the whole, and you need to take random samples from the whole area. Now here we've got a big Quadrat. You can count the whole thing, or you can just decide to count one of the smaller parts within it. Now you do not throw a Quadrat. That is a very, very common mistake in exams. If you've got a Quadrat, you can either put it randomly in the field by closing your eyes and just dropping it, or you can use a random number generator to say where to put it using coordinates that you would have on a map. You need to take repeated samples, and you need to remember that if this is your Quadrat, it's 50 centimetres long and 50 centimetres wide, then that's a half and a half, but when you multiply it, that's actually only going to give you a quarter of a square metre, so you need to be careful when you're sizing up the larger area. Here's a practice. Now this is a patio, and actually a patio almost works like Quadrats because each square on the patio is like an individual Quadrat. You've got a key down here, you've got ladybirds, spiders, worms, fairly self-explanatory. But if you look at it, you can see depending which Quadrat you sample, you get a different idea of what is living in the garden. If you took this Quadrat, you think there's one ladybird, one spider in this area. You took this quadrat, you think there's just worms. What you have to, you can probably spot it already, is this worm here goes across 2 quadrats. So you'd actually have to decide how much of a bug would be in each square before you counted it. Same with this spider, that's across 2. Do you think the body's here, so that's where it counts? Doesn't count here because the legs don't really count. This worm, would you choose all of it? Would you choose both of it? Anyway, you're going to have a look at this. You've got some questions coming up about it, so if you've downloaded this file, you can go backwards and forwards to help you answer the questions. Otherwise, you might want to pause the video here and copy the pictures. Now, here are the questions, and we've got our little calculator friend there to tell you you're going to need a calculator. How many of each type of organism live on the patio? Not on an individual quadrat, on the patio, all together. So you're going to have to add them up. Calculator could be handy. How big is Jay's patio? You'll have to count the number of squares. It does tell you. Each slab is 1 square metre, so you've got all the information you need there. Jay's next door neighbour has a patio that's twice as big as Jay's. Estimate how many of each type of organism might live there. You can't be sure because there might be different soil, there might be different levels of moisture, but you can estimate. It's not a guess because you're not choosing random numbers. It's going to relate to the answer for question 1. Question 4, what is the average number per paving slab? That's a mean. Remember, you're going to add it all up and divide by how many slabs there are. I'm not telling you how to do that. I'm sure you can do it. Number 5, estimate how many of each organism would be found on a patio made of 50 slabs. Quite a lot of maths in this, so when you write it down in your books, ready to send me a shot, or you're putting it into your notebook on Teams, please make sure you include your working out. Then if you've made a mistake, you will spot it, or I will spot it. Hopefully if you make a mistake, you will spot it first and change it, and then I can say well done. Now, what we're going to do at home is you are going to have a go at estimating the population size of a plant species using random sampling. This is the first half of the required practical that you would do at school. You've got 2 options. On the left-hand side, you have got sampling in your own garden, if you've got one, or a park, if you've got one nearby and your parents have given you permission and you're able to follow social distancing rules. Before you start, you're going to need to make a quadrat of your own. I would suggest using the back of a cereal box, making sure the cereal is empty first, making sure your parents don't have any other plans for the box. Then use a ruler to make a frame shape and cut out the centre. If you need a plant identification sheet, print one of those. I will have one loaded up for you to help you in the documents. Randomly place your quadrat and sample. You're going to have to repeat it 10 times. The next slide does have an example of a table you should draw in advance to make it easy to record what you find. You don't want to be trying to remember it all. If you are not able to access a safe outside space, either because you haven't got a garden or because you're social isolating for yourself or somebody else in your house, I'm also going to provide a quadrat template and some pictures that count as mini fields. You're going to cut out the centre of the quadrat, so you've got a mini quadrat. Place your quadrat on your field, record how many of each plant type, repeat it 10 times. Okay, so let's have a look at the table now. Quite self-explanatory. It says results. You've got space for 10 samples. You've got space down the bottom for mean. You've got plant A, plant B, so you can just look at 2 different plants. You might decide to do daisies and dandelions. I wouldn't suggest you do grass because it will take you a long time to count grass. You might have other weeds in your lawn. You might have clover. As I've said, I put a sheet and you have a look. I can't judge what you're going to find. If you go into a park, you might get slightly different plants again. You are going to need your calculator here because you have to find the mean for both sets of plants. This can be done with the printed sheets as well because there are 2 different plants on those sheets. Last bit you've got to do is answering 6 questions. Again, you're going to need your calculator. Told you there's quite a lot of maths in here, but that's good because we like maths and we are good at it. So, which plant had a larger population? What area did your quadrat cover? So work out the area of your quadrat. Remember, that will be inside the frame, not around the outside, because if you go for the external measurements, part of that will be covered up by the actual frame itself. Roughly how many of your quadrats would it need to make a square metre? And remember, because a metre is 100 centimetres by 100 centimetres, it's actually 10,000 square centimetres in a square metre. What advantage does sampling have? Rather than me sending you out to count all the plants in your garden, how could you improve accuracy of your answers? What would happen to your results if you changed from a shady area to a brighter one, or the other way around? So some quite interesting questions here. Obviously you can take it further. You can decide that you actually want to do something similar with watching birds in your garden, seeing whether are there more sparrows, are there more blackbirds. The only thing we need to be a little bit careful of at this time of year is that lots of birds are nesting, so they will be moving about quite often, and it can be quite tricky to tell if it's exactly the same robin or blackbird or sparrow that you saw 5 minutes ago. Anyway, I hope this makes sense. As I said, there are some extra identification and support sheets with this activity that will all be loaded on Go for Schools and on Teams. Of course, as always, you can email me with any questions as you go along, as well as sending through your work at the end. Don't forget, there's also a Seneca task going up shortly. Take care. Hope to see you soon, Year 10. Bye.